everyone who attended yesterday had a great first day. I know that I had a very good experience, a lot of useful discussions. So just wanted to give you a little bit of primer for day 2. Oh. Oh, I see it. Okay. All right. So beginning the day, we're going to continue with our parallel sessions. The first session will be in vitro studies that may contribute to alternative bioequivalence approaches for locally acting orally inhaled drug products. And the presenters you see here will be giving talks on the topics of morphology and other in vitro approaches. The second session, which we'll be continuing from yesterday, will be in vivo and in silico studies that may contribute to alternative bioequivalence approaches for locally acting orally inhaled drug products. And the presenters you see here will be giving talks, and these talks will be about the use of modeling to inform PK metrics on regional deposition and regional deposition validation. And we're going to conclude the day with session 3. So the idea with session 3 is that we're going to take everything that we've talked about, coming back to the first symposium, which was about the comparative clinical endpoint and pharmacodynamic studies, and then session 1 and session 2, which are talking about various techniques that may be useful for an alternative bioequivalence approach. So more of the technical details. And then this third session is going to be a series of panels. It's really going to be 2 panels with more or less the same panelists in each panel. And we're going to be talking about how do we take all of these techniques, put them together into what might be a useful alternative bioequivalence approach. So you can see the presenters that we have here and the moderators that are going to be running these panel discussions. And these will be the panelists participating in the first panel. And after this, we'll have a break, and then there'll be a second panel with these panelists that you see listed on the screen here. And concluding the workshop, we're going to have closing remarks from Dr. Bing Li. And that will conclude our workshop. So thanks again for coming today, and I hope you have a great second day. Seems like most people have settled in. So I'm going to start with some introductions. The first speaker today is going to be Dr. Kevin Feng. He is a reviewer in the Division of Quantitative Methods and Modeling and Office of Research and Standards and OGD FDA, same division as me. He started in April 2019 as a reviewer, and prior to joining FDA, he worked in Certara with over 13 years of experience in quantitative clinical pharmacology at Farsight, which is a Certara company in North Carolina, and also translational drug development in Simcyp, which is a Certara company in the UK. He has extensive knowledge in application of preclinical drug development and clinical drug development, also including but not limited to new and generic drug application submission and regulatory review, managing grants and contracts, and acting as grant and contract officer for communicating with internal and external stakeholders. Before joining Certara, he worked for 2 years in finance modeling on a portfolio optimization project with the Smith Institute in Oxford, UK. He got his PhD in 2004 in automatic control and system engineering from the University of Sheffield in UK. Our second speaker is going to be Dr. Jgen Bulitta. He is a professor from the Department of Pharmacotherapy and Translational Research at the College of Pharmacy at the University of Florida. So we have some Gator supporters here. He is supported by the University of Florida's Preeminence Program in Drug Discovery and Development, and he received the Perry E. Foote Eminent Scholar Chair Endowed Professorship in 2019. Dr. Bulitta has 20 years of hands-on experience with population PK and mechanism-based modeling and has significantly contributed to 97 phase 1 or phase 4 clinical PK studies. Also, he has been working on inhaled drug PK together with Dr. Gunther Hochhaus, who we have here today, for over 7 years. A third speaker is going to be Dr. Jan de Bakker. He is currently the CEO of Bulitta. He graduated from Delft University of Technology in the Netherlands as an aerospace engineer and earned his Master of Science degree in aerodynamics and specialized in applied biomedical computational fluid dynamics, leading to a PhD from the University of Antwerp in Belgium. He is an alumnus of the MBA program at the London Business School, London, and the Columbia Business School in New York. He has received several awards for his innovative research in the field of airway modeling in respiratory and sleep medicine, and his work has been published in international journals. He founded Fluida in 2005, and he has held the position of CEO since 2007. And our fourth speaker today will be Dr. Ching-Long. He is the Edward M. Mielnik and Samuel R. Harding Professor and Departmental Executive Officer of Mechanical Engineering Department at the University of Iowa. His research focuses on developing computed tomography, image-based subject-specific lung models, employing machine learning and deep learning of CT lung images to identify imaging clusters and advance a cluster-informed modeling strategy, conducting CT and SPECT human subject studies for model validation. And Dr. Lin received his MS and PhD degrees from Stanford University. He received the NSF Career Award and is elected Fellow of the American Physical Society, the American Society of Mechanical Engineers, and the American Institute for Medical and Biological Engineering. So without further ado, I think we can welcome our first speaker, Dr. Kevin Feng.
Good morning, everybody. I think luckily I am the first one to talk about this topic. I think we have a very good discussion yesterday. I really enjoyed the discussion. Oh, something not moving. Oh, okay, it's fine. I can use the mic.
Oh, okay.
Yeah. So the topic, I look at the slide. The topic is slightly different. So the title is acceptability of using alternative PK metric from system PK data to inform regional deposition for oral inhale drug product. So I'm from the quantitative clinical pharmacology team from DQMM, ORS, and OGD. So here's the disclaimer. This presentation reflects the view of the author and should not be construed to represent FDA views or policy. That's a general comment. I was going to do a few of the outlines of the introduction, talk about what is the challenge that's already been talked about, and then looking at what kind of research that's already currently available, and then looking at an example on the in vitro deposition study and its relationship with the PK. And then I'm looking at another example on the normalization on drug dose to inform the relationship between the PK, and then looking at the population PK and the partial AUC as alternative PK metric for the discussion that we had already had yesterday. We're looking at the partial AUC, and then I show you one of the ground opportunity, how the ground will be provides opportunity to do the further research and provide a summary. So the challenge that we already know that we have been talking about yesterday, the whole day about the challenge, the weight of evidence, the challenge, and also the clinical endpoint, and the PK that is really challenged because the highly variable and a lot of huge amount of subject. And another one is because also in the modeling, the high PK variability can mask the underlying PK metric different when the regional deposition was used to identify the formulation difference. So the research that many research are ongoing, some research are investigating the feasibility of assessing formulation difference in regional lung exposure based on system PK concentration to establish PK BE or inhalation drug product in lieu of clinical endpoint studies. So here's one of the example. This example is really that FDA granted FDA with this example for the in vitro deposition experiment. So in the experiment, that's using the next generation impactor. So the assumption is looking at the flow rate as like, for example, 39 liter per minute. And then there's a 8 chambers or 7 stage. And you're looking at the particle size, it comes from one end to the other. And then you're looking at the particle size on the mass difference. And you're looking at the different stage. And at the last stage, stage 7, the particle size a lot smaller. So first stage is a lot larger. And then we're looking at for the job, we're looking in particular for Bulitta zone and South. And then you're looking at because we create different batches of a difference for the different formulation particle size, and you're looking at the significant difference between all of these batches, and looking at there's a premise A stage, and looking at the flow rate as 80 liter per minute for 4 seconds, and looking at on the left there's a Bulitta zone, on the right is a South. There's a provide significant difference with in vitro and in vivo difference. And then we also have another ground we're looking at to build up the model using the for example the Bulitta zone, and this Bulitta zone they have a double peak. If looking at the double peak of the systemic PK data, it's really that the model is trying to looking at the peripheral airway and central airway, and to capture all of the information from the system PK model. There's a pop PK model that was also mentioned by Rose yesterday, about how this population PK can be used. However, although you can fully capture all of those PK phenomena and double peak, and then looking at the in vitro on the right, on the in vitro deposition. But the in vitro deposition, you're looking at a stage, and there's also a lot of variation. How can we link to this peak in vitro and in vivo together? So that's some of the questions. And with the same ground project, we found that one of the information that we're looking at the correlation to build up the correlation about how to using the parameter of the different size of the parameter. There's on the x-axis, there's 4 different batches, like R1 is the reference and T1 the test. There are four different reference because there's also the variation between the different particle size. And then you're looking at the correlation with the EUC, and there's with the fire particle size is more likely to deposit to the peripheral round, and we identify like one of the 1 micron and 2 micron. There's a positive correlation can be found, and really this example is to show that from the population PK point of view, we can identify how the in vitro correlation with the in vivo PK metric. However, this is a scientific investigation. The aim is to looking at the correlation between the difference of the in vitro and in vivo, but we cannot use this example as a guidance. Okay, we can use the in vitro data for the decision making. So this is one of the example that we have been working on. And this another example that FDA did, and FDA did a lot of research. This example is really to looking at we create a lot of in vitro experiments that looking at the difference between the dry powder formulation, that the different type, including like the MMAD, the different size for 4.5 and different size, and also vary the dissolution rate. So some are higher in distillation rate, the other one is lower. And then looking at the in vitro total lung, that was normalized to determine the average dose passing through the 3 anatomical mouse throw models, and the other normalized PK total lung factors to looking at how the total drug can be deposited into the lung. So the aim of this example is to looking at the various way to interpret the data to understanding how various in vitro data have impacting the in vivo PK study. So this example, even looking at this example, the significant PK profile you can see can be generated from the different in vitro data. You're looking at a lot of variation from the different in vitro diffusion, also from the different mouse throw model. And looking at the data set, because we normalize data on the left, we say, okay, those normalized, they're using normalized factor. On the right plot, at the before normalization, if you look at before normalization, because those amount are different. So and then the AUC to infinity, they provide different results for on the left after those normalization is for the AUC is in BE, but on the right before normalization the AUC is not BE. However, the Cmax is both of them are non-BE, even you normalize. That because the particle size different, dissolution different. So those are experiments. It show to you that some of the deposition would have some of the difference. So it's really this example is really the idea to show how we can integrate this different in vitro method together with the PK difference to represent the clinical significance use the local lung concentration at the site of action, and to identify what is the most significant method is for drug to deliver the site of action with the combination of PK data, including the in vitro data, particle size or dissolution data. And here's another example. It comes from also from one on the ground, also come from the end of submission as well. So this example is really to looking at to build up. We already have the PK data. The PK data is equivalence. So it built up the model using the central run and peripheral run, and then looking at two compartment model, and they validate the model and create the model. And when the model was created, like central and peripheral, however, in looking at the total, in looking at the B sim after do the model was created and do the simulation, I found that the total lung is equivalence, of course. And then looking at central and peripheral, they are not bioequivalence. So that is to show that even if the PK is a bioequivalence, but you're looking at central or peripheral, is not equivalence. And then also looking at absorption half-life. So one of the fast absorption and slow absorption for the one of peripheral fast absorption, then the half-life is really short and only 3 to 5 minutes or so, really a few minutes. So it's really very short. So this example is really to investigate the PK relationship with the simplified empirical PK. So and this information is the question is to whether the way of analyze the PK to inform the local drug concentration and site of action in addition to the drug deposition data. One of the possible way is to build up a full long PBPK model, and with the CFD deposition method, which is enabled to shift the particle size, physiological value, and also you're looking at the different the deposition ratios. So and then we can looking at mechanism model and simplify the model and to see what information and what kind of impact can we get. So this is example is really tell you that one of the example may not be sufficient. So to looking at the information for oral inhalation drug product. And then you'll be looking at the model in terms of model, when your model is validated, and on the left, it tells you how the model will be sensitive. So on the x-axis is the C to P ratio. There's a central to peripheral, the ratio between those, and you will vary about the difference. And then you're looking at the partial AEC because we already know that there's a very rapid absorption. There's a few minutes, and okay, how can we looking at the impact on a partial AEC? Like even looking at the 5 minutes, 10 minutes or 20 minutes, can we use the partial AUC as an alternative PK? That we have a discussion yesterday as well. That we say, oh, partial AUC, it is one of the information we can use. And this information, okay, if we can use, then looking at the PK analysis, then the like 10 minutes and 20 minutes, they are really equivalent. So the question is now, we already know that the C2P ratio is sensitive. And then can we use the partial AEC to do for the TBE as alternative method? But however, if looking at on the left graph, the C2P ratio is from 0.4 to 0.5. That's correlated with the data from 80% to 125%. And this C2P ratio is really one of the model-derived parameters. Okay, and it's really different from the true physiological values. So can we trust of this C2P method? So, and also in addition, because if looking in the C2P ratio and also the partial AEC, and especially for the slow and the normally for the Cmax, for the slow and the slow release and slow absorbing drug, they may be variable, but for the most of the drug product is very rapid dissolve and very rapid absorb. So it's really that they create a lot of high variation at the very beginning for the Cmax. So can we? The question is, can we use the partial AUC to replace the Cmax? And also because the information, but we need more information to get whether this information is sufficient, such as the in vitro information. How can we use the value to inform the data? So is in here is really that we're looking at how this the data can be used. So we are encouraging the people to provide. At the moment, the FDA is really looking at how this information, how the partial AUC can be used, and what the kind of information can be used. So if any question and any information, FDA will encourage to provide some of the to submit us as a pre-ENDA meeting, like the Rob has mentioned yesterday in keynote speech, and contact us. We are really happy. Come very early on, you will provide information that we can discuss, and in the hope that we can finalize this partial AEC as alternative method, then we can use this method to provide the information, either put in the PSG or any guidance for the future development. So here is looking at how the acceptability of the alternative PK metric to inform the regional deposition. So definitely the partial AEC-based approach on system PK may be possible, but for this information, more detailed information should be provided to support the approach, including the appropriate validation or rigid. And the approach should be sensitive enough to detect the formulation, like the C2P ratio. How sensitive the method can be used, and also to demonstrate how the clinical significance using a proper metric and such as AUC. I also provide some of the graph on the right, that the CFD model and the PPPK model, and it really it may be more likely to achieve success with a combination of the top-down and fully mechanistic approach that used for the PPPK, and that is at least including system compartment and regional deposition parameters. So from this example, it shows that both of the empirical simple PK model may be good for certain drugs and can provide some of the general solution to support alternative PK metric. The alternative way to a simple PK model is to develop more mechanistic approach. The power of the mechanistic model can be more flexible to understand the effect from the various factors, such as to shift the PK particle size, or to change the physiological value, and to revise the deposition ratio So both of method have their own strengths in terms of scientific approach. We encourage both of the approach, but the goal is the same. So how do we interpret the system PK data and use the data in a modeling framework? So and then in summary, that for the empirical model can provide generalized solution such as using the alternative PK metric PAUC, while the mechanistic model can provide further support such as using the in vitro data, dissolution data, in vitro deposition data, CFD models. And then we integrating all of this approach that like provide step by step and looking at further investigation. So here is one of the example, the grant with current grants that we are available. The available is really that to understand that we have been talking about early on, that is really on the first step we create a more complex physiological model, and with the physiological model, and then we can simplify the model. After the model was built, then using the pop PK model to see how the population PK model can represent to the physiological parameters, so that we give us the information and impact on different drug property or different structural physiological parameter that we can use, and then we can use those to summarize how the key factor can be used. So this is one of the available grants that we're currently available. And in summary, that there exists a challenge on conducting the comparative clinical endpoint and for PD-B study for oral inhalation drug product. And the modeling and simulation could provide a solution as alternative PK metric, such as PAUC from the system PK to inform the original drug deposition and to show the equivalence in local site of actions. However, sufficient justification and should be provided. It is not as a show as a empirical and the partial AUC, and we will come up with a solution in after we have conducting some information. FDA really support innovative approach to support the approval of the oral inhalation drug product, and also the different funding we also available for any university or institute or even the industry they can apply. So here is the acknowledge on the team. Yeah, that's my presentation. Thank you.
So good morning. My name is Jgen Bulitta, and this presentation was created together with my colleague, Dr. Günther Hochhaus at the University of Florida. This is on the feasibility of predicting regional lung exposures from systemic PK data of generic OIDPs via population PK. I would like to acknowledge our collaborators at the University of Bath, at VCU, McKinnell, and at Worldwide Clinical Trials for the bioanalysis. So after inhalation, a certain amount of drug deposits in different lung region, and this gives rise to systemic plasma drug concentrations. Another portion of the drug can, of course, be swallowed and then enter the gastrointestinal tract and be absorbed. For the purposes of this presentation, we assume that there is no gastrointestinal absorption, either due to low oral bioavailability or due to charcoal block. However, we will look into extensive detail into the benefit of having available intravenous PK data and how to handle them within a pop PK analysis. The overarching question is, can a pharmacokinetic study assess pulmonary bioequivalence? What does population PK add, and how can we implement pop PK and its variability components properly? So specifically, the three most important questions are, what is the dose available to the lung? This is available both through non-compartmental analysis AUCs or partial AUCs, or by population PK modeling by the amounts of drug deposited in central and in peripheral lung, AC and AP. So here we are well covered. The second question is, how long does the drug stay in the lung? This is accessible either by peak concentration or by mean residence time from NCA, as well as by the pulmonary absorption half-life from pop PK. And then the third and perhaps most tricky question is, what is the central to peripheral drug deposition ratio in the lung? In certain situations, this may be accessible through Cmax, as Dr. Feng has outlined in his presentation before, but this is really where population PK shines if a drug has biphasic lung absorption, we can nicely estimate the individual subjects' amounts of drugs deposited in central and in peripheral lung. Now, the hypothesis and model here for fluticasone propionate is that for a slowly dissolving drug, PK should allow us to assess differences in the pulmonary dose, the pulmonary residence time, and then in the regional lung deposition. So the idea here is that we have a certain nominal dose, which is deposited in the central and in the peripheral lung region. And then mucociliary reclearance only affects and removes drug from central lung, but not from peripheral lung. And so we have here two parallel first-order absorption processes and then a three-compartment PK model. For fluticasone propionate, oral bioavailability is low and was neglected in this study. We didn't use charcoal and we did not have IV reference data. And as of last night, literally, this publication is available in PubMed and under this link online. So the goal of this original study was to probe whether PK is sensitive to differences in the C-to-P ratio for slowly dissolving FP. Our collaborators at the University of Bath, Jack Schur and Rob Price, created 3 fluticasone propionate formulations which were aimed to have the same dose, the same dissolution rate, but which differed in their central to peripheral lung deposition ratio. And this was achieved by modulating the lactose spikes while using the identical powder for fluticasone propionate. When different in vitro studies were performed to characterize the formulations and eventually a human PK study, a crossover was performed. So the EPSD deposition profiles here show that on stages 2 and 3, the 3 formulations, even though they differed quite a bit in the MMAD, the stages 2 and 3, the amounts deposited in those larger particle stages, are somewhat similar, suggesting that the mucociliary recurrence may have affected the 3 formulations in a similar way. In contrast, on stages 4 to 7, it was very clear that the larger formulation A had a much smaller amount of drug deposited here. And when using the Preludium software, the central to peripheral ratio was clearly predicted to be more on the central side for formulation A compared to B and C. When Dr. Hochhaus's lab performed 2 different dissolution methods, so the USB paddle method, where formulation A, the larger one, clearly dissolved more slowly compared to formulation B and C. And in the transfer system, where the time frame here is a lot longer, which is possibly more physiologically relevant, formulation A again was much slower than formulations B and C, with the mean dissolution times being 15 hours, 13 and 10 hours for the larger and the 2 smaller formulations. And this is published in detail. In the PK study, we performed a 4-way crossover double-blind study in 24 healthy volunteers with the 3 formulations. And formulation C was repeated in order to get access at the intra-subject variability. The total dose was 500 micrograms FP, given by 5 individual capsules of 100 micrograms each. LCMS was excellent and performed at worldwide clinical trials in Texas. And then, of course, we performed non-compartmental and compartmental population PK analysis. So, as Dr. Feng has already kindly shown, the 3 formulations here had nearly identical terminal phases, so the half-life was identical and independent of the formulation, 10 hours. But formulation A, this is before dose normalization, has clearly lower peak concentrations and also lower areas under the curve. So this was very informative data. Then we performed a population PK analysis, and this is the scenario which we published, where we did implement IV reference data from the literature. This was from a study by Torsen et al. We also tried another study by Allen et al. So the individual curve fits, so we observed versus fitted concentrations were nearly perfect here. So this looked really good. But there was a slight bias, not terrible, but a little bias for the population fits. And we could show that this is because we borrowed the IV disposition parameters of occurrences and volumes of distribution. We borrowed those from the literature. When we didn't borrow anything from the literature, it was still variable, but the bias went away. So we considered this the more conservative approach because it guarantees that we have the literature values for the alpha, beta, and gamma disposition half-lives, and so we published the left side. The visual predictive checks here for formulations A, B, C, and CR showed very nicely that this central tendency and also the between subject variability of the observations for each of the formulations were adequately captured by the population PK model. Now, we also tried, both by the original data set and in a simulation estimation study, to compare a model with 2 versus 1 pulmonary absorption process. And so in 200 out of 200 cases, the model with 2 pulmonary processes clearly won. over the one absorption process model, and this was by a very large margin. So this, if a drug is biphasic lung absorption, PopPK very clearly could identify that it was biphasic. No, not a close call whatsoever. Now, how do we implement the population PK results into the bioequivalence testing for pulmonary drug deposition? This is done at the individual subject level parameter estimates, so the so-called post-hoc estimates, where when we compare the total dose deposited both in the central plus the peripheral parts of the lung, with 3 formulations, each compared to CR, were bioequivalent and point estimates close to 100%. In contrast, the dose deposited in central lung for formulation A was clearly above 1 and also not bioequivalent, and the dose deposited in peripheral lung for formulation A was clearly smaller than the dose from formulation CR. And then that gave rise to a similar difference for the C divided by C plus P deposition ratio. So very clearly, the individual subject estimates could distinguish between formulations A and then B and C, whereas B, C, and CR were all bioequivalent for all metrics. So this meant that population PK could inform bioequivalence testing, and it could distinguish between the central and peripheral lung regions at the individual subject level, and that is a big strength. Now, the population PK parameter estimates, so the population means, we used a literature estimate for mucociliary clearance of about 8 hours half-life. The half-life of drug absorption from central lung was 9 hours for larger formulation A and about 7 hours for formulations B and C, nicely correlating with the transverse system data. The absorption from peripheral lung unit is minutes here, so it was 14.6 minutes for A and about 7 minutes for formulations B and C. So again, a clear distinction here, and clearly absorption from peripheral lung was much faster. And then with regard to the drug deposition into central lung, formulation A had 62 micrograms versus 12 micrograms into peripheral lung, so very centrally depositing. And then formulation B and C was approximately equal with regard to central versus peripheral drug deposition. So then we took those population estimates and ran Monte Carlo simulations to simulate hypothetical formulations. So these are very similar to formulation A from above. So A is a very centrally depositing formulation with a 0.83 C divided by C plus P ratio. Formulation B and C are identical, and they both have a 50-50 deposition centrally versus peripherally. And then formulation D was a more peripherally depositing formulation with 0.31 for this ratio. The absorption half-life from central lung was taken to be 7 hours, and from peripheral lung 7 minutes, as for formulations B and C above. This may differ a little bit between formulations, but we simplified at this point. I would like to point out that the alpha half-life from IV drug disposition was 13 minutes, so close to the 7 minutes, and the gamma disposition half-life was 10 to 11 hours, again, similar to the 7 hours here. So now we threw a lot of dice in our random number generator. So we generated a 5A crossover study with formulations A, B, C, and D, with 500 micrograms FP inhaled each, and an IV 10-minute infusion 80 microgram FP reference dose, which gives about the same AUC. 400 subjects each were simulated with between subject variability and between occasion variability and different covariate effects. And then we randomly generated 100 bootstrap data sets of 24 subjects each and estimated all of them under 4 different conditions and approaches. So the first approach could be considered the gold standard. In this one, we have individual subject IV reference data and use them in the population PK model. Initial estimates were reasonably okay. So this is when you can inform at the individual subject level the alpha, beta, and gamma half-life, which is a big strength. The second approach has a perfect population PK model in the same population as you have in your own bioequivalence study for inhaled drug. But here we only inform the model at the population mean and between subject variability level. We do not provide individual subject level data. So this is the second best thing you can do. And then in approach 2, we fix the alpha, beta, and gamma half-life from IV drug disposition. In approaches 3 and 4, in contrast to 2, we again don't use IV literature data, and we only fix the population mean clearance in order to estimate the drug deposited. That is good, but in approaches 3 and 4, we did not fix at all the alpha, beta, and gamma half-life, and instead tried to estimate that based on inhaled drug PK data. Approach 3 was a little bit more careful with regard to the initial estimates, but the approaches were somewhat similar. Now, how do they perform? Amounts of drug deposited in peripheral lung. Here you are looking at the means from 100 population PK estimation analyses. So these are population level data. So formulation A for approach 1 here, the red horizontal bar and the other colors represent the true value. So A is perfect, B and C are perfect, C is 5% underpredicted, but so approaches 1 and 2 here very nicely capture the true values. So bias with the ideal value being 0% was very close to 0 and certainly not statistically significantly different. Slightly worse were approaches 3 and 4. They were not far off for the amounts of drug deposited peripherally. And then precision, which is the standard deviation of those point clouds, was better for approaches 1 and 2. But overall, peripheral lung deposition, very good. Central lung deposition. So here, the models could very clearly distinguish that formulation A was a centrally depositing formulation and formulation D was a peripherally depositing formulation. Nevertheless, for formulation B, we had a 15% bias for these approaches 1 and 2. It was not a statistically significant bias, but it still is not as good as I would have hoped it should be. Nevertheless, we could clearly distinguish between the 4 different formulations with regard to their C divided by C plus P ratio. And the precision was best for approach 1 and a little bit worse for 2, 3, and 4. I would like to point out that for this study, we had to use settings for 400 estimations at the same time. If this was a single data set, I would have personally definitely gone into full Gargamel mode and found out how to get rid of this 15% issue here. Nevertheless, the results were still quite good with regard to C, central and peripheral deposition. Now the absorption half-lives, basically perfect results for approaches 1 and 2, really small bias and larger, but better precision for approaches 1 and 2 versus approaches 3. Approaches 3 and 4 were a little bit biased high, maybe not statistically significantly so, but they were not perfect. So here 1 and 2 are best. And then the absorption half-life from central lung, the ideal value being 7 hours. Here the 4 different approaches perform very similar, basically very little bias at all. And precision was comparable between all 4 approaches. Now, when we look at bioequivalence testing form, it is very important to not forget about the individual subject estimates. So this is a data set with 48 subjects, so a little bit larger, where we look at the ratio between the individual subject estimated divided by the true individual subject value for each of those disposition parameters. So here that means that for all of the subjects, the individual subject estimate was very close to the true value with a very tight confidence interval of plus minus 5%, the ideal value being 1.0. So approach 1 kind of hit it very well. Approach 2 had a little bit of an issue here for V1, probably not dramatic. The confidence intervals were a little bit wider, but this is where approach 4, where we estimated the alpha, beta, and gamma half-life without having IV data, kind of had about a 10% bias at the individual subject level for 3 out of the 6 parameters, and the confidence interval was wider. So here, approach 1 clearly was best. Now, when we look at the arguably most important results of this presentation, this is the individual subject level estimate versus, so the estimated versus the true C divided by C plus P ratio. So here, formulation A, which had a true value of 0.83, was very nicely distinguishable with centrally depositing formulation, very nicely distinguishable from formulation B, which deposited more peripherally. So here the true value is about 0.3. And when formulation B and C were supposed to be identical, it didn't come out perfectly identical, but it's still quite close. And the true mean value is 50-50, so 0.5 here on the x-axis. So here POP-PK could very nicely distinguish between the different formulations. And it is these data which are most important for bioequivalence testing. Correlation coefficient was 0.95. So in conclusion, POP-PK is a top-down approach, as Dr. Feng has already said, which could very nicely distinguish between the centrally and the peripherally deposited doses in lung. And it could inform ANOVA and BE testing in both lung regions. Generating individual subject level IV data in the same study is recommended if possible. The next best thing is to have fixed population PK parameter estimates for the drug disposition, so the clearances and volumes of distribution based on a literature study. Estimating the model population PK model for inhaled drug without IV data performed reasonably or adequately but tended to have a couple of additional intricacies and was less robust. Usually precision was a little bit worse. The amounts deposited both in peripheral and central lung could be estimated robustly. The absorption half-lives were a little bit less precise. And of note here, the between subject and between occasion variability for those half-lives was relatively large, as we showed in our paper. So therefore, we propose that non-compartmental analysis based Cmax may be more robust to assess the rate of absorption compared to these absorption half-lives estimated from POP-PK. Overall, it is our opinion that non-compartmental analysis, POP-PK, PBPK, computational fluid dynamics, and other mechanistic approaches are complementary, and we should leverage each other's strengths. Of course, this also feeds into the mechanistic in vitro approach, is not part of this presentation. Now, the results presented here are technically limited to fluticasone propionate, but we would like to propose and perhaps speculate that this approach is also applicable to other slowly dissolving corticosteroids. Clearly, future research is needed. Population PK modeling could overall provide valuable insights into the regional lung deposition for drugs with biphasic absorption, and these insights are not accessible by standard non-compartmental methods. So I would like to say a big thank you for all of the people and co-workers who very hard worked on our clinical study and the modeling. Dr. Hindle and co-workers at VCU, Dr. Schur and Price from Bath, Worldwide Clinical Trials, FDA colleagues, and ultimately funding from FDA. Thank you kindly for your attention.
Good morning, everybody. It's great to be here in person. It's great to see everybody. And it's great to have the opportunity to discuss what we've been doing the last several years in trying to better understand the regional deposition of inhaled drugs and also the regional exposure to the lung. And then we've been talking a lot about these in silico methods, and also the term digital twin has been going around quite a bit in recent years. I would say it's driven by the AI hype and all that. So there's more emphasis on it than ever before. But in order to really trust these digital twins, we have to be sure that they adequately represent the underlying physiological processes. And I think we know very well that computational fluid dynamics, which has been around for decades in other industries, it has the possibility to really understand flow behavior and particle behavior. But we also know that it very much relies on the accuracy of the input parameters. So we have to make sure that the input parameters are accurate, but it also has to take into account the variability that we would expect to encounter in the real world situation. So in our case, can we include not only the correct input parameters, but also an adequate level of variability that we can expect in a patient population that we're trying to assess? As Brian nicely showed yesterday, there are several key parameters that drive deposition of inhaled drugs. We have the device, we have the drug formulation, the inhalation profile, so how the patient uses the device, and then also what we think is very important, the lung structure and the lung function. So if you look at how to approach it from the digital aspect, we can make very accurate computer geometries with the device. So we can very clearly include the geometry using these computer models. As we've discussed at length, there's a whole range of in vitro characterizations that can be done to really understand the properties of the formulation. For inhalation profiles, we increasingly measure how patients are actually using the devices, whether it's a DPI and MDI. So that's also something which can be digitized and then used in the in silico approaches. And then to understand lung structure and function, we use what we call functional respiratory imaging. So we've been developing functional respiratory imaging, or FRI, for the last 17 years or so. And it starts from the conventional CT scan, so the low-dose CT scans. And what I think is one of the biggest shortcomings in modern medicine is the fact that radiologists typically look at your CT scan for about 2 minutes. And then they visually assess it and they summarize it probably in 2 lines. And then after a few months, they delete the scan. So in my opinion, they probably extract 5% of the information that's available in the scan. So what FRI tries to do is we convert these 2D CT slices into 3D reconstructions of the anatomical structures. So we look at airways, we look at blood vessels, lung volumes, lobe volumes. So you visualize it, but you also quantify it. And you keep the numbers, you don't throw them away after a few months. If you have inspiratory, expiratory scans, we can make static images come to life. So we can look at regional ventilation patterns, airway resistances, but also the deposition of inhaled particles, as you very well know. So just by using the inspiratory, expiratory scan, without the need to radio label any other formulation, we can quite adequately assess where those particles end up, where they deposit on a patient-specific level. And so we continuously validate this. So we've compared this quite extensively with methods like the scintigraphy. So there's an increasing body of literature where the FRI parameters, so the CT-based parameters, are compared with results from the scintigraphy. So that's pretty much on a total lung level. So that's total lung dose. But as we also discussed in several topics, or in several presentations, SPECT is one of the alternatives that also provides more regional information. So a while ago, we published the comparison between CFD and SPECT-CT in asthmatic patients. And you can see that the correlation is quite good. The agreement is about 1 to 2%. And the reason for such a good agreement is that you indeed include the relevant parameters that drive deposition, that drive particle transport. You have the patient-specific ventilation, you have the patient-specific upper airway geometries. So that really provides confidence that what you see in these in silico models that are fed by the patient-specific data provide results that you could also expect to see with the more advanced clinical tests, in this case, the SPECT-CT. So FRI provides quite a comprehensive set of parameters that provides insights into the lung diseases and also the deposition of these inhaled drugs. So why is it important? So if you look at some of these diseases, you can see that the structure changes quite a bit. So on the left-hand side, we see a healthy volunteer. But if you look at, for instance, a typical COPD patient, you see hyperinflation. You see those lungs are significantly larger than the healthy subject, where the airways are significantly obstructed. So you see narrowing of those airways, which obviously will affect regional deposition, regional ventilation, and so on. In asthma, also an obstructive disease, you see also a level of hyperinflation and a level of bronchoconstriction, which can be reflected by the FEV1, but as we discussed yesterday, I think FEV1 is somewhat useful, but also has many, many flaws. Cystic fibrosis, I think Will touched on that as well. You see clearly bronchiectasis, a level of hyperinflation. IPF, you see traction, bronchiectasis, restriction of the lung volume. So you typically see a reduction in overall lung volume. So a large level of heterogeneity that we can expect in these lung disease patients. If you look at the more functional components, so what you see here is the regional ventilation in an IPF patient compared to a matched healthy subject. And the color codes indicate the change in lobar ventilation. So you can see that those lower lobes are red. They're relatively underventilated compared to a healthy subject. And the upper lobes tend to compensate, so there's more air going to the upper lobes versus the lower lobes. So ventilation changes on lobar level in idiopathic pulmonary fibrosis. Also in COPD, we do see changes. So again, you can extract this from this inspiratory, expiratory CT scan, where you can see that if you look at a healthy subject, the difference between upper and lower ventilation, so the box plots barely overlap. But in a COPD group, you see there's more overlap. So you do see shifts again from upper versus lower and vice versa. It's important to also include the device. So we all know quite well that these devices create different type of flow patterns in the upper airway. So here on the left-hand side, you see the turbohaler. So a lot of turbulence generated in the mouth region versus a typical MDI, which provides quite a different flow pattern. And obviously, these flow patterns influence particle transport, they influence regional deposition. So quite important to also include the key parameters of the devices that we are dealing with. Importance of upper airways. A lot of patients, a lot of people in general have either very small upper airways or large upper airways. It's related to their BMI, it's related to the presence of sleep apnea. So you can clearly see here that if you look at these concentration maps, people with more narrow upper airways tend to have a higher degree of deposition in those upper airways. So a very important part to also include those upper airway characteristics if you want to have a digital twin that is representative of the clinical situation. And so one of the lessons for me for the last day or so is that I think there's a need to bring it all together. I think there's tremendous expertise in a lot of these subcomponents, in vitro testing, PK testing, CFD, etc. I think we're at the level where there's a lot of value in bringing the expertises together. I think it was also mentioned in the previous talk is that most of them are complementary, but it's also important that, I think Ross mentioned that in his talk, that the output of one stage also becomes seamlessly the input of the next stage. And that's why we started to work together with Nanopharm to develop what we think is a smart track approach, where indeed we're attempting to seamlessly let the phases flow into each other. And just to give you an example, so this is a comparison between two of the devices, two of the triple components or triple products on the market, Trimbo versus Trelegy. And that's where we did the typical in silico approach. So we went to our database that's been built up over the last 17 years. We selected 20 COPD patients with associated clinical parameters. So with an FEV1 range of about 20% predicted to 66% predicted. We looked at the recommended flow rates for use. So for a PMDI between 60 and 68, for a DPI between 30 and 90. So we looked at different inhalation flow rates to see what is the variability that we could reasonably expect in the real world situation. Then, as we showed before, we couple the devices. So for each individual airway model, upper airway, lower airway model, the devices are coupled, both the MDI as well as the DPI. And then we work together with Nanopharm to look at the aerosol particle size distribution as a function of the inhalation profile. So you clearly see that the MABD, GSD, fine particle fraction, they differ as a function of the inhalation profile, but they're also quite different between this DPI and the MDI. So again, an input parameter for the models. And then the results show pretty much here on a deposition level, intrathoracic, peripheral deposition, where do we see overlap, where do we see differences? And then the box plots, they represent the level of variability that you can expect in the patient population, in the inhalation profile, and so on. You can also represent it like this, where it's more of an equivalence assessment, so the 80 to 125 limits. So you can see, okay, for the subcomponents, do we fall within the limits or are we outside of it? So these obviously are not two products that are supposed to be equivalent. So I think the drug developers have their own reasoning for using larger or smaller particles. So you do see that there is a good way of distinguishing between the deposition patterns induced by these typical devices. So we've worked together with Omar Osmani to do this work, and we published it a few years ago. Of course, visualization is interesting. So you can see that typically for these extra fine particles that you see in Trimbo, so the Chiesi product, they tend to go to the more deeper lung, whereas the larger particles, especially I think in this case, the inhaled corticosteroids, they stay more in the central regions. So I think CFD is a very valuable tool. It's something that is quite accurate, but also, as we've discussed before, it takes a long time to do these simulations. And that also means that you're usually limited in the number of patients and the number of scenarios that you can do. So what we did, and it was quite recent, so I think in the last months, last year or so, we developed what we call the rapid deposition analysis, or RDA. So as we mentioned, we have quite a large database of these CT scans. We've run over the last decade or so thousands of CFD simulations. So we went back and we tried to identify what are the key drivers for deposition. So with an AI-based approach, including non-dimensional analysis, we were able to come up with an algorithm that still allows to assess patient-specific deposition patterns that are inhaler agnostic. So we can use the technology to assess MDIs, DPIs, soft mist inhalers, and so on, where the accuracy is still quite good. So we have an accuracy compared to SPECT and CFD more than 90%, Having said that, the RDA does not include the more advanced physics, the multi-physics flows, evaporations, etc. For that, we still need the CFD, but the advantage is that it's much faster. So where typically for a full CFD simulation, you require several hours. You can now do that in a much shorter time frame. So this is about 30 minutes if it's a blank CT scan, so where you have to extract all the features of the geometry. If you have the features of the geometry, it's literally a matter of minutes, seconds to do these type of scenarios. So what that allows us to do is to use this rapid deposition analysis to kind of mimic what you would do in a very large clinical trial. So include a lot of patients, include a lot of variability, run thousands of scenarios to really understand what is the broader field, what is the broader possibility of scenarios that we can encounter. We can then use that as a basis for a sample size calculation and then follow up with the more advanced computational fluid dynamics to include evaporation, the physics, the actual geometries of the inhalers, etc., such that at the end of the day, you have an in silico approach that reflects as closely as possible the true clinical situation that you might have when you do actually perform a clinical trial. Because it's on patient data, it includes the variabilities that you reasonably could expect in a clinical situation. And then if you have the regional exposure, you can feed it into the PK models and then look at absorption and do the remaining of the pathway of the smart track pathway that we're trying to develop. So in conclusion, we discussed the key drivers for deposition, the devices, the drug formulation, the inhalation profile, and very importantly, the structure and the function. I think we can say that we can adequately include them in these in silico approaches. And I think with advances in the technology, you can start using AI and non-dimensional analysis in a very complementary way to go beyond the limitations of purely, let's say, the computational fluid dynamics. So the RDA plus the CFD, we believe, provides a good pathway to really have an in silico approach that mimics what you can expect in a clinical situation. And I'm quite excited about this and about this approach in generics, because we see that a lot of these sponsors today probably have a better understanding of what their generic drug is doing, what the journey is from the particle from the device all the way into the lungs, probably better than the reference, probably better than the originator, simply because they're encouraged also by the agencies to use these technologies more and more. So I do think there is a huge possibility or a huge opportunity also to increasingly use it for new chemical entities, to also develop better devices, better formulations, and really move more towards what I would say the more precision medicine, the personalized medicine. So I think the future looks bright. So thank you very much for your attention. Looking forward to the discussions later today. Thank you.
Thank you, especially Ross and the workshop committee for this opportunity to give a presentation. I feel so good to have an in-person meeting. And today I'm going to talk about cluster-informed in silico and in vivo regional deposition assessment. I'm sorry about the kind of the aspect ratio of the slides different. As a result, some of the fonts are kind of overlap. So what's cluster? Cluster is also referred to as subpopulation, subgroup, or subtype. And in this presentation, the CFPD, P stands for particle. CFPD predicted particle deposition will be compared against CT SPECT measurements. We like to apply the machine learning, deep learning to CT images to identify imaging cluster in asthma, COPD, post-COVID patients, and then compare particle depositions between subjects in different disease, and then try to explain why the deposition patterns are different between subjects and across disease. So this is the outline of my presentation. I'm going to talk about briefly CFPD subject-specific blood model, and then quantitative CT imaging variables, machine learning and deep learning, and then cluster-informed strategy. Then I will apply CFPD and quantitative CT to asthmatic. COPD, and post-COVID. So our subject-specific city-based CFPD model consists of several steps. First, we use image one from full inspiration to segment lobes and airway, CT resolved airways. Then we use these two to construct the four one-dimensional conducting airways. Then we register image one with image two at expiration. Then we can derive regional ventilation information. Then we combine structural and functional information together for our CFD simulation. And we have 3D and 1D. 3D can be applied to regions of interest, so we can get detailed simulation information. 1D is for the whole lung. So our model essentially couple 3D and 1D simulation. And for our 1D model, we are able to predict particle depositions in conducting airway, respiratory airway, and also alveolar on inspiration and expiration. For details, you could refer to our paper. So what's quantitative CT variables? At full inspiration, we are able to derive several metrics, including branching angles, circularities, wall thickness, and airway diameters at different airways. For instance, trachea, left, right main bronchus, and then two segmental airways. And then on expiration, you can see that we are able to derive functional variables such as air trappings. If we register inspiration and expiration images, then we can further classify air trap voxels into small airway disease voxels, as you see on the right in yellow. FSAD stands for functional small airway disease. And then red means emphysema. So that means we can use registration of inspiration and expiration scan to further identify FSAD and emphysema voxels. We can also use registration information to derive the regional for distribution for the whole lung at voxel levels. And also Jacobian means deformation. And then ABR means tissue deformation. We can also use the image data to calculate the lung shape, because for some lung disease, lung shape is an important metric too. So you can see we have variables at local level, at global level, and low regions, including structural and functional variables. Then we apply machine learning and deep learning to these variables to identify imaging clusters. So here is the summary of the cluster-informed strategy. First, we identify imaging clusters in patients with different lung disease, and then apply our CFPD models to cluster subjects and try to understand why the deposition in different subjects between different diseases are different. And the strategy is multi-scale because it can bridge population and individual scale. We also bridge global and regional scales. To give you an idea, let's look at small airway disease. Small airway disease small airway by definition means airway diameter is less than 2 millimeters. You look at the distribution of a small airway in the lung, they kind of reach maximum around 6 and 7 generations. And let's look at the lower right-hand corner. We compare small airway in the healthy subjects and the COPD subjects. And green means normal voxel. And then yellow means small airway disease. Red, emphysema. If you look at the cross section of the small airways, you can see that in healthy subjects, is more circular than those in COPD. Okay, in COPD. Now let's look at the cluster information between asthma, current smokers, and post-COVID. Let's look at asthma first. We have 4 clusters with increasing severities. We found that only cluster 4, severe asthmatic, has very severe small airway disease in red. For current smokers, you can see small airway disease, FSAP, appear in all clusters and increase with severities. And for post-COVID subjects, only appear in 1 cluster. There are 2 clusters for post-COVID patients. So we are going to use that information to explain why the positions in different clusters between different diseases are different. So let's first look at asthma. We analyzed 106 non-severe and 142 severe asthmatics. For details, please refer to our paper. We used the previous quantitative image metrics for clustering, and we identified 4 clusters. So you can see, I show all the important imaging characteristics. Then we established association between imaging and clinical characteristics. So you can see the corresponding characteristics between imaging and clinical. And what's important is cluster 3 and 4 are both severe asthma, but they have quite different structural and functional characteristics. Small airway disease only appear in cluster 4, but not cluster 3. That means if we use PFTs, both have very low PFTs, but small airway disease only appear in cluster 4. And I will show you that the particle depositions are different. Okay? In addition, you look at the right-hand side. Cluster 3 is predominant female versus cluster 4, male. So we conducted a pilot asthma study funded by FDA, supervised by Ross. Because of funding restriction, we only have recruited 6 male subjects because cluster 4 are male dominant. So we compare results from cluster 1 and cluster 4. We recruited 3 subjects for cluster 1 and 3 subjects for cluster 4. We acquired this image data, and the particle size is less than 1 micron. Then we co-registered SPECT and CT images. So here is kind of a result of co-registration of SPECT and CT lung images. On the left-hand side is the mild asthma. Right-hand side, severe. Here we define coefficient of variation. It's the ratio of standard deviation over mean. And we use CV to quantify heterogeneity of particle deposition. So you can see, for instance, on the left-hand side, CV is 0.43 versus 0.58. You can see with severe asthma, we have local hotspots compared with mild. There is no clear local hotspots. So let's look at the 6 subjects that we recruited. And target C1 means we use PFTs, FEV1 or FEV1 over FVC criteria to recruit these subjects. So you can see these 3 subjects versus the other 3 subjects, they are quite different in terms of PFTs. However, if we use the previous cluster information, we design a simple cluster model on the right-hand side. That means we only use 4 variables to classify any new subjects. As a result, you can see the 3 subjects in cluster 1 belongs to cluster 1, determined by imaging variable. But then for severe asthma, only subject 22 have very severe small airway disease. Okay. But then the other two severe asthma do not belong to cluster 4. Here I use simple cluster means I use S cluster means I use simple cluster decision tree to classify these subjects. Okay. So let's compare CVs between these subjects. If we use target clusters, so for instance, target cluster includes subject 2, 4, 5, and cluster 4 includes subjects 20, 21, 22. And the CVs are essentially the same. We cannot see the difference between the two. And there are two columns, CT spec versus CFD. Okay. And we also use healthy as a reference, but we don't have CT spec data for healthy subjects. So we could not see the differences between the two. However, if we use imaging cluster information to compare the results, then you can see simple cluster 1 consists of 4, 5, 2, 4, 5, and simple cluster 4, unfortunately, we only have one subject, 22. And you can see the CV is higher than cluster 1. Okay. So I hope that we have, in the future, we are able to recruit more subjects to verify the difference between the two. Okay. And then here we compare the lower particle distributions between simple cluster 1 and simple cluster 4. Again, simple means we use simple cluster decision tree to classify these subjects, as opposed to using PFTs. Okay. And you can see the local distributions are quite different between cluster 1 and cluster 4. And there's so much information here. For instance, you look at the first two columns, blue and orange. They are both cluster 1s, but one is from CT spec, the other from CFPD. The agreement between them are pretty good. However, if you compare simple cluster 1 and simple cluster 4, you could see the differences, especially on left upper lobe, left lower lobe. You can see the change in distributions. And so is right middle lobe. So you can see how sensitive our 1D CFD model is and how the distributions change due to the lung severities. Now let's look at COPD. We have analyzed former smokers and current smokers. You may wonder why we separate former from current smokers, because smoking status is a confounding factor on CT numbers. So we have to separate these two. In addition, we like to consider the biological variable effect, especially age. And we found that the former smokers tend to be older than current smokers, which seems obvious. You look at the data. In fact, they are quite different in age. And that's why we separate former smokers from current smokers. And I have to emphasize that we use cross-sectional data. We also have the result from longitudinal data. So we call that longitudinal clusters. And these two kind of clusters are different. One depends on severity. The other depends on disease mechanism. So we found that the imaging metrics for the cross-sectional and longitudinal clusters are slightly different. And that's very important. You want to kind of understand the depositions in smokers. So for this, we recruited 14 COPD patients at 0 and 12 months. 0, 12, and 24 months. So it's a longitudinal 3 time points. unfortunately, only 8 subjects completed all 3 visits, and 5 were former smokers, and 3 were current smokers. And look at the gold stage severities. You can see we have 2, 1, 4, 1. We don't have large samples. We also acquire PFT, CT, and SPECT images. Now let's compare CFD and CFPD predicted CT and SPECT results. So on the left-hand side, C stands for current smokers, F former smokers. And you can see C1 and C3 are almost the same. And same is for F1 and F3. But then if we compare current and former smokers, 1 and 3 are different. In addition, you look at the former smoker cluster 4, F4. You can see the CV is very high. So that means there are local hotspots, especially in F4 subjects. Then we can also look at the right-hand side. We compare COPD with asthma. You can see COPD has overall higher CV than asthma, because small airway disease is very important in COPD, but less important in asthma. And that's why you can see the CVs different between the two. Finally, let's look at post-COVID-19. The reason we did not have in-person meeting over the last 2 years is because of COVID. So we only have the CT data, but we don't have CT spec. So here I'm going to show you the imaging cluster for post-COVID and also CFD predicted results. So we use 140 subjects. And these subjects were enrolled between June and December 2020. And these subjects, we have data at initial visit and about 4 months after recovery from infection, 4 months, okay? We also have a control group, we call that cluster 0 control, healthy control. Then we identify 2 clusters. One has small airway disease versus the other fibrotic lung. And you can see there's so much information. I'd like to point out that air trapping or the small airway disease in the middle part. That cluster 1 has small airway disease. And it's kind of equivalent to cluster 1 in COPD, initial stage of COPD. And what's unknown is whether small airway disease in cluster 1 here will progress over time. That's very important because in COPD, small airway disease progress to emphysema. Or we don't know whether cluster 1 here, small airway disease, will progress or not. No one knows. Okay. Another important feature in post-COVID is ground glass opacity, and we are able to capture that as well. So we use our CFPD to predict CVs, and you can see cluster 0, control group, cluster 1, and cluster 2. And cluster 1 tend to have higher CVs because of small airway disease. And we also found that cluster 1 has increased airway resistance. As for cluster 2 has reduced airway resistance, and particle depositions in cluster 2 is also smaller than deposition in cluster 1. So all these need to be also validated against CT spec data in the future. In summary, the CFPD predicted coefficients of variation agree well with those measured by CT spec. And also shows that the cluster-informed strategy could explain the differences in particle deposition between subjects across disease. Thank you.